are we ready? No. Friday. Yay. Okay, we're ready. We got lecture capture going. All good. I'm going to get some negative supercoiling right here. Okay, so we're going to continue with replication, and then don't let me forget, before the end of class, we need about 5 minutes. There's a little practice exam question in a quiz, which probably doesn't require respondents because I'm assuming I probably set it up wrong. But I don't care because that's going to be one that you'll be able to go back and look at, right? So you'll get your score and you'll be able to see it forever and play with it. It's just an old exam type question so that you can get an idea of an application type question that's not just, you know, a lot of the stuff on the quiz is what does primase do? What does this do? What does that do? You know, I did a couple more like I would write on the exam for this quiz to try to get people have a better idea of how I ask questions. Because normally by the third exam in a class, you know how the teacher asks questions. But since now I'm not Dr. Stein and we do have essentially completely different styles for exams, right, suddenly it's like the first exam again. And so I give you a setup, I'm a protein that does all this crap, who am I, is more like the things that I would ask, rather than what is the function of primase, or what is the function of ligase, or, blah. So more like, if you know the function of ligase, or helicase, and primase, and topoisomerase, those questions shouldn't be a problem for you. It's just students aren't necessarily used to reading that kind of question and immediately going, oh my God! But you know the answer if you know the definition, right? You just have to think a little bit backwards from there. And so then the little quizzy thing at the end, and it's just a free point, right? So it's not a real quiz, it's not one of the drops, it's just, I hope I only made it a point. I hope it's not 100 points. If it's 100 points, it's not really, don't get excited or panic, right? But just a little extra point. And there was an extra point on today's quiz because last quiz had shitty questions, the heritability baloney, and then there was the one without the 5', 3', and I didn't realize Stein gave a rule about that, and so that made my answer wrong. And so that will help us get points back for some shitty questions on the quizzes. All right, so at the beginning, we talked about the origin of replication, right? In Procs, there's one. In Eucs, they're all along the chromosome. At each origin, we go both ways, right? The fork opens both directions. That's bidirectional, 2 directions from every origin, right? So it doesn't just travel to the right, to the right, to the right, or to the left, to the right, off of both, on both sides. And the first thing we have to do is set up a way for DNA polymerase to come in and start adding our deoxyribonucleotides. And remember I said DNA polymerase is anything that's classified as a DNA polymerase cannot add to naked DNA. Okay, so if a bacterial DNA polymerase cannot add to naked DNA. Right? A eukaryotic DNA polymerase cannot add to naked DNA. It has to have an existing nucleotide sitting there in order to do it.
Can you elaborate on the phrase naked DNA?
Yeah, I'm just going to do that. So naked, meaning just the template and nothing else, right? So if I draw our template, and we'll sort of make it like a little bit of a bubble, right? And these guys are still, and remember, the black lines are just nucleotides all together. The red lines are the hydrogen bonds between those. And then, right, each strand has a polarity, and they're anti-parallel all the time. And we'd say our origin is somewhere down the middle, right? So the proteins bind, opens the bubble, and helicase is sitting right there, breaking hydrogen bonds, go, go, go. So this is a snapshot of replication at an instance in time, right? Again, it's just a process. It doesn't really stop. Ooh, I have a good-sized bubble. Let's fill it in, right? It's just helicase is continually going in both directions, but if we take a snapshot to see what's happening, that would be what this is. It just, we stopped it and we did some freeze frames, stopped it in time. Oops, that's an ORI. Okay. So these template strands would be something I'd call naked, right? There's no existing nucleotides. If I draw some just to help us, I'm just going to draw A's because I'm that lazy, right? And then what would this side be? Ooh, complementary base pairing, right? So the line is really just a whole bunch of nucleotides. The other line, I just don't write them out. Okay? So I would call this naked. There's nothing there. DNA polymerase can have all the deoxyribonucleotides its little heart desires, and it cannot do diddly squat. Okay? Because it requires an existing 3'OH waiting for it to add something to make a phosphodiester bond. Okay? So I was trying to explain it this morning, and I know Dr. Stein talked about enzymes and protein conformation. She loves conformational change, right? Did she not get frothy when she said conformational change? Right? Got all splash zone in the front. And that enzymes are super, super specific for one reaction, one bond, and a very specific bond. So DNA polymerase creates phosphodiester bonds. 5' phosphate, 3'OH. The ligase enzyme, which we're going to talk about later, also makes phosphodiester bonds. 5' phosphate, 3'OH. So why do we need ligase if they're making the exact same bond, right? Enzymes make one specific bond. Why can't DNA polymerase just start adding onto naked DNA or just the template strand? Why do we need ligase at the end to fuse Okazaki fragments? Why can't polymerase just do that? It's the same bond as phosphodiester bond. If we go ahead and look at those bonds after the process is done, you cannot tell who made it, polymerase or ligase. It's the exact same phosphodester bond, condensation reaction, lose water. Looks exactly the same. Yeah. Yes, so exactly. So ligase can fuse two existing nucleotides that are sitting next to each other. DNA polymerase cannot do that. DNA polymerase has to have a nucleotide coming in to an existing. So I like to show it like this. Here's the nucleotide. Here's the 5' phosphate's made into a phosphodester bond on this side. There's a bunch of nucleotides here. Here's this. Ooh, that finger doesn't want to do that. Ooh, that one's not. Okay. So this is the 3'OH here, right? Here's a 5' phosphate, a 3'OH. DNA polymerase makes this bond, and then hydrogen bond. So its active site fits only when an existing nucleotide is sitting like this and an incoming one is like this. That's its shape. Okay? If two are sitting next to each other, like this, DNA polymerase cannot grab those two and make the phosphodester bond. It's not the right shape for its active site. The active site of ligase fits that and does that phosphodester bond. Ligase can't fit like this, can only fit like this. Okay? So it's all about enzyme specificity, why DNA polymerase is a big loser and can't just make that. We had to have another enzyme do that, because it can only bring them in. So it has to have this existing 3'OH. When this is sitting here, there's no existing 3'OH, right? Because the 5' phosphate and 3'OH are already made into phosphodester bonds, right? All of those on the templates, that's how they're connected together. The backbone. So there's nothing that DNA polymerase can do. The enzyme primase has the ability to stick, make it another color, make it green, a nucleotide to hydrogen bond onto that naked DNA, right? Can just start that reaction. But it can only hold on to ribonucleotides. Its active site only fits ribonucleotides. That little change in chemistry from a ribonucleotide to a deoxyribonucleotide The deoxy, right, missing the oxygen on the 2'-OH, those do not fit in the active site of primase. Only ribonucleotides do. So he can put that one there, take that one 3'OH, do a phosphodiester bond here, add another one, phosphodiester bond, add another one, phosphodiester bond, and after 15 or so nucleotides, you know, I'm representing that as 4 of them, primase is done and falls off. And this guy now has an existing 3'OH hanging off, right? The little pinky's hanging off. Now, DNA polymerase can bring in a deoxyribonucleotide, make the phosphodester bond, and hydrogen bonding pops it up onto the complement. Yeah? Yeah, sorry, I'm doing it backwards. Here, let me change it. Thank you. This is really 5 and this is 3. I wasn't paying attention. Don't do that. Pay attention. Okay, do you guys see what she was saying? Remember our rules, has to be anti-parallel, has to be complementary base pairing, and we can only add the 3' ends. So I was being a big doofus, and I was going this way. I was traveling off of 3'OHs, that's good, right? And I was complementary base pairing, but what rule did I totally fuck up? Yeah, anti-parallel. So to fix that, I just changed the polarity of the strand, right? So now this is the 5' end, that's anti-parallel to the 3', this is 3', anti-parallel to this guy, right? Anti-parallel to this guy. Thank you. See, I did that on purpose just to, no, I didn't. And so then I'll start adding some, I'll do the DNA guys, just so then I can make my next little point. Ooh, sorry. Go back. No. So right, here's the 3'OH, and we will, actually, we'll use black. We're going to now add deoxyribonucleotides, so what does A complement to? T. Right, so all these guys are going to be added. This is the 3'OH here. We have the phosphodester bonds between them, and the phosphodester bond between that uracil and the thymine, the ribonucleotide to the deoxyribonucleotide. Why can we do that? Why is that okay? How can DNA polymerase make a phosphodester bond onto a ribonucleotide? Because its 3'OH looks exactly the same as a deoxy, right? If we go back and look at chapter, I think it's 4, I've been lying about 3, right? The 3'OH is exactly the same on both. It's the stupid 2' that's different. 2' has diddly squat to do with phosphodester bonds. That's necessary for its overall chemistry and whether primase can hold it or not and blah, 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 but nothing to do with phosphodester bonds. So DNA polymerase doesn't know or doesn't really care whether it's a ribo or a deoxyribo, as long as it has a dangling 3'OH for it to grab onto. Right? So it's the same phosphodester bond. Again, if we just looked at the chemistry of that, condensation reaction, lose water, looks exactly the same. No different. Okay? And then, again, once we get there, remember this was just a snapshot. Really, the fork is continuing to open, and that one just kind of follow. As it opens, polymerase just keeps adding and adding and adding, so it either falls off the chromosome or bumps into, right, the bubble. If we do like a, this would be a close-up if we zoomed out and we say, this is the one we're looking at, right? Ours is going like that. This one's going like that. That one's going like that. So these two here are going to end up bumping into each other at some point. These two are going to bump into each other. This one just falls off, and that one falls off, if that's our fake little chromosome. Okay? And so these guys will end up bumping into an existing primer sitting there from the lagging strand of another bubble. But we'll get there in just a second. Okay? And so if we're, yeah, go ahead. Let's get to that right now. So the question was, which of these is the Okazaki fragment? None yet. Because as this opens, right, as we break more hydrogen bonds, we just keep adding, and we're just following the fork, right, as it opens. That's called the leading strand. The new strand is the leading strand, not the template strand, right? The leading strand is the newly synthesized strand. The template strand is just the template strand. And in DNA replication, both strands are a template strand, okay? So there's no Okazaki fragments on the leading strand. There's only Okazaki fragments on the lagging strand because we still have to obey the rule of anti-parallel, complementary base pairing, and adding to 3' ends. So if, pretend like we have a primase that's got a developmental delay, and let's say the primase adds a primer like this. Right, we've got complementary base pairing, phosphodiester bonded, and in order to be anti-parallel, this has to be the 5' end of that nucleotide, and this has to be the 3' OH of that nucleotide, okay? And on this side of the origin, going that way, we want to fill in that side of the origin, okay? So I like to tell students, and it's a lie, but it will help you, I promise, and I'll tell you the truth. The origin is the no-fly zone. Do not cross the origin. Don't cross the origin. Don't do it. You cannot cross the origin, okay? When I'm asking you to fill in a replication bubble on the exam Monday, you have to remember anti-parallel, complementary base pairing, add to 3' OHs, don't cross the origin, okay? So you're not allowed to cross the origin. We're going to hold on to that for a second. Let's finish our lagging strand, and then I'll come back to why you can't, okay? Right now, we're going to make it a memorized rule so you don't forget about it and go crazy on the exam and go the wrong way. So that stops you from going the wrong way. So you can do process of elimination, right? If you have a stupid bubble and you're freaking out and you can't remember shit, right? And you're given a polarity and you're like, holy crap, people, and I'm told I have to fill in the right side. If I fill in the left side, I get 0 points. I have to fill in the right side, so I can't just memorize one thing and then just regurgitate it on the exam. I have to understand this process to apply my rules, the 3 rules, and don't cross the origin. So all you have to do is say, okay, I'm going to do the experiment. I'm going to put both of my primers at the origin. We know one of them has to be at the origin, okay? And then all we're going to do is set up our anti-parallel bullshit. We're going to remember, don't freaking cross that. So you're going to draw a big fat line, don't go that way. And now we're going to decide which primer's in the right place and which one's in the wrong place. Which one do we use, the top or the bottom? The top, because there's a stupid 3' end, and we fill in this, and now we're anti-parallel, we have complementary base pairing, we added the 3' end, blah, blah, blah. This one's traveling in that direction. If this was the fork, we know that guy is leading. And then we go, okay, well, if he can't be here, he must be over here at the fork. 1 primer's at the origin, 1 primer's at the fork. We set up our anti-parallel baloney. We pretend like it's complementary base paired. We're just drawing lines because we don't want to spend our time drawing a million letters. And then we extend off the 3' end, and we stall at the origin, right? And actually, I'll make it so that we know is our little primer guy. Or I'll make it a box, because I'm going to ask you to make the primer look like a box so it's clear which line is the leading strand, which one. And then this one happens to be the lagging strand. Well, how can you tell, and what the hell is this Okazaki fragment BS? Right? So I haven't even gotten there yet. I'm just talking about lagging strands. Yeah.
Wouldn't it be discontinuous?
Yes, it's discontinuous. It's lagging. It actually takes longer than the leading strand. And so we only notice that it's lagging. I mean, you can memorize lagging always goes towards the origin. Leading always goes towards the fork. Okay? I also like to refer to the fork because it doesn't really look like a fork. Right? A fork in the road, I don't know, that makes me feel like, you have an option to go both ways. Like this is just, it's a crotch. Okay? The leading strand goes right into the crotch. Okay? Who doesn't want to go into the crotch? The leading strand goes right in. Right? Hydrogen, open up the crotch, leading strand goes right in. Okay? Lagging strand, kind of the loser, going back to the origin. Not very exciting at the origin. You can't even cross the origin. Go back to the origin. Okay? And it does take longer because, watch when this magic happens. We're going to move this over here. Because remember, that was just a stupid snapshot in time. Right? Both of these crotches have been opening the whole time we were busy with primers and complementary base pairing. Right? So that was a snapshot. If we then wait 5 seconds or 10 seconds, whatever, and take another snapshot, right, this strand is now way down here. Here's the fork. The leading strand, just helicase, right? We'll just remember our little friend. We'll call him helicase. Right, is breaking hydrogen bonds opening. He's not waiting around for primers to get set up and complementary, but he's just going. And so is polymerase, just adding to the leading strand, following right behind helicase. As soon as there's a broken bond, boom, polymerase is adding the complement, right behind helicase the whole way. Right? Well, what about the sappy little lagging strand? What's his problem? He can't just follow the crotch, because he has to go the other way. So if we took let's pretend like our next snapshot in time was right here. That's where the fork was, the timeline from left to right. Then primase had to sit down another primer at the crotch. Polymerase goes back and bumps into the next primer and then falls off. And then the next snapshot in time, oh my God, the crotch is already open this far. Hell. Primase sits down the primer, DNA polymerase adds on, bumps into the next one, falls off. Right? The Okazaki fragments are the primer plus its strand, because the primer is phosphodiester bonded here, right? Between the purple box ribonucleotide and the first deoxyribonucleotide green guy, that's a phosphodiester bond. This is one piece of DNA. I'm calling it phosphodi. Okay? That makes sense? Yes? From when we set it up, right? But when this guy bumps, what about right here? Is there a phosphodiester bond between that last one and that primer? No, because DNA polymerase can't do that, because now we've got 3'OH and 5' phosphate sitting next to each other. Polymerase can only do this, cannot do this. So it stays as a gap. Okay? And, what the hell? This is RNA. We don't want RNA in our DNA. So we don't want a phosphodiester bond to that ribonucleotide, because we want to get rid of that bad boy in the next step anyway. So Okazaki did the experiment, and that's why they named it after him. He froze a cell during replication, looked at one chromosome, and remember how I said heat or boiling can break hydrogen bonds, right? So he heated up this stuff, and then looked at the fragments of DNA. Ran an electrophoresis gel, separated by size, whatever. And what he found was 2 strands that were ginormous. Those were the template strands, right? And then he found, I'm going to draw with the, a bunch of strands that were pretty long. And now remember, he's looking at multiple bubbles all the way through, right? So the leading strand is not the same length as the original template strand, right? The template strand is one whole single-stranded DNA from an entire chromosome, cajillion base pairs long. Each leading strand from each bubble could be 10,000 base pairs long. Pretty freaking long, but not a cajillion base pairs the size of a chromosome. And then, I'll draw in the other direction, even though you couldn't tell the direction, just to help us. He found a whole crap load of little fragments. What are those? The Okazakis. He said, "Holy shit, people!" I don't know if that's what he said. He was probably super nerdy, so he probably said, "Oh boy." And so, because the biochemists had looked at these enzymes and said, DNA polymerase can only add to 3' ends. How the hell do you make replication work? Right? When scientists just thought we did what makes sense, right? You have this is the entire huge cajillion base pairs, right? You do that, and you do that, right? One long strand, a cajillion base pairs long replication on one side, one long strand, a cajillion base pairs long replication on the other side. If that's what's happened, then all you'd see on your gel is that and that. No. They saw this. And so that, the hypothesis of how replication happened to account for all these stupid little fragments makes sense by the biochemistry that we knew. DNA polymerase can only add to 3' ends, has to be anti-parallel, and that a primer has to set up each one. Okay, so that's the stupid Okazaki fragment. So it's the primer and the lagging strand attached to it, covalently bonded, right? Phosphodiester bonds are covalent bonds. So heating the shit out of it will break hydrogen, but will not break covalent. You could boil the living crap out of DNA, you will not break those phosphodiester bonds. But the hydrogen bonds are blown to crap. Yeah? That's a good question. Yeah. So we're going to get to that, but his question is, so when you say you're not a rabbit, what do you mean? When they bump into each other, right, the leading strand coming one way from a bubble ends up bumping into a lagging strand going the other way, right? Because if this is the origin here and this is the origin, this guy is leading into the crotch. But this guy from the other bubble is leading into the origin. So a leading always bumps into a lagging on this side, and then the leading from the other bubble bumps into another lagging from the other one. And again, at those points, there's no bond because DNA polymerase can't do the existing crest. Yeah. No. So they're not, exactly, that's a good question. So at each place where an Okazaki starts, it's just part of that, right? The origin is right in the middle and we go both ways, and the lagging strands coming back this way, right, are all just wherever, when the fork opened up enough, primase sticks another primer, polymerase comes back and fills in. By the time that's done, oh shit, now it's time to put another primer, fill another primer, until there isn't room because a leading strand the other way is now in your way. So yeah, there's multiple Okazakis per origin. And so I showed 3 or something, right? Now I can't even figure out what the heck I'm doing. Here, where? Yes, right there. Right? These are 3 Okazaki fragments. So I will ask you to include 3 Okazaki fragments on your drawing, and I want you to circle them exactly where they are. Okay? And so be neat, because messiness ends up getting you a 0, because I can't tell if you're just messy or if you're being cheesy. Okay? It's that cheese ball factor that runs around, because it's the primer and the lagging strand coming off of it. This is an Okazaki fragment. This is one piece of DNA, RNA hybrid, right? But one nucleic acid, all phosphodiester bonded together. Like I said, between, right there, there's no bond there. So, unfortunately, let's try another color. Students have done, well, the ones that do this, right, they're like, well, it's a lagging strand and a primer, but it's not the right one. That's not one piece of nucleic acid, because there's a gap in between. So that's definitely no, wrong. That's just, like, wrong, right? But cheesy is when they do this. Which primer do you think goes with that lagging strand? Were they just messy and did a quick circle? But all 3 of them are like that. Okay, 0. I can't tell. I can't know what's in your head, right? I can only grade what's on your paper. I don't want to think people are cheesy, but unfortunately I've run into plenty of cheesy people in my life, so I have to, right? If you're neat, there's no issues. Okay? There's no way, if you're neat, I'm going to say you're cheesy and you're faking it, right? Okay, if 2 of them are exactly right and 1 overlaps a little, you're fine, right? You don't have to, like, spend 4 hours making this little circle. But you do have to make sure it's clear that you know what you're talking about. Yeah?
So there's no bond at this fork, so that's where the characterization is.
Yeah, so there's no bonds any place between the Okazakis or between the leading when it bumps into the lagging. So that was just if, I don't have enough colors. I'll try gray. So this was a snapshot in time. That was when the fork had opened to there. Remember, it's a continuum. And then this was where the fork was open when the primate dumped next to the crotch. And then 10 seconds later, it was open enough, it dumped another one. And then 10 seconds later, it's open enough, it dumps another one, until it runs into a leading strand coming from the origin over here. Does that make sense? Yeah, so it's just like taking snapshots in time. Really, this is all just a big continuum, but for us, we kind of need to do that. Okay, so now let's go back to the do not fly zone. I know this is super messy. And I said oh yeah, sorry, go ahead. Okay, hold on. If I'm not, then make sure you ask me. So the do not fly zone, the do not cross, is because it's bidirectional. And as soon as the origin snaps open, whatever, breaks hydrogen bonds, opens up enough, the primers sit at the origin on both sides at the same time. Okay, so it's not like we'll do the right side first, then we'll do the left side, right? I have to draw one side first and then the other because I can't simultaneously draw 2 things at the same time, right? I need Dr. Stein here, I'll do the right side, she does the left side, and we race to try to get it exactly the same, So really, when this bad boy is stuck here from primase, this bad boy is stuck here from primase. As soon as the origin's open and there's enough room, boink, those guys both sit there. This is the 5' end and the 3' end, right? Because it's anti-parallel, complementary base pairing. And then our good friend DNA polymerase can start adding phosphodiester bonding nucleotides. This is now our crotch on this side. It continues opening. Crotches should. And this guy will continue following. That's the leading strand on this side of the origin. So the lagging, stupid Okazaki bastard that I drew over here can't cross the origin because there's a primer sitting there. But when you're drawing, there's not going to be a primer sitting there unless you pop both at the origin at the same time. So if you just remember don't cross, you can't go the wrong way. You can do your process of elimination, add to the 3' end, don't cross the origin, and get it all correct. But you just can't cross it because there's somebody sitting there, right? This leading strand, there's nothing to complementary base pair if it tries to, right? These are already hydrogen bonded down here. This strand can't come up there. There's, right? It just plunks here, and there's no bond here. So there's no bonds across the origin either. That's another place we have to deal with, right? This one would be the lagging strand, right? In this snapshot in time, we set up there. Polymerase adds, point, don't cross the origin. Well, it can't because it runs into the primer on the other side. So this is a gap where there's no phosphodiester bond. So gaps at the origin, gaps between Okazaki fragments, gaps between where bubbles combine, where bubbles bump into each other. Okay? And at this point, we're still left with all this stupid purple RNA. Has anybody ever told you that there's RNA in our chromosomes? No. So we have to get rid of it before the end of S phase so that our chromosomes are now pristine again, DNA and proteins, right? That's it. No RNA is in there. We got to get rid of that crap. Yeah. So it doesn't know. That's good. Good targeting for primase at the origin is likely controlled by the initiation complex. Yeah, it always does it because that's the only way it can complementary base pair and have the 3' end going the correct way. So there's got to be some targeting mechanism, protein-protein interaction that helps guide it. At the actual crotch, it's got to be targeted by proteins too. And it's all of this I'm taking completely apart. Physiologically, it's really not what it looks like. We're keeping everything separate to understand the process. But really, all of this kind of happens in one big, huge protein complex with DNA running through it. So all of these things are targeted by both complementary base pairing and all that stuff, but protein-protein interaction, so they know where to go. How the hell else? It doesn't just know, right? So it's all targeted in that way. Yes?
On the left, you want us to also add the protein?
No. So we're not going to draw any proteins on the drawing. It's all going to be nucleotides, right? So the protein stuff will be covered in the multiple choice, more like the quiz questions. Because otherwise, I won't be able to tell what's the box primer, and like at the fork, we'll have, well, here's helicase, and then we've scribbled all over, I can't read 3', 5' ends. So it'll say don't add any enzymes, don't add any proteins to it. Okay? Because then it's just, it'll be too messy. Won't be able to see polarities. I'm going to ask you to tell me polarities and all that stuff. So this whole drawing of the first part of replication is in that tutorial, Malone Replication Part 1. Okay, so if you want to go ahead and watch that without all the questions and without me doing all my extra, BS crap that I do in class. Right, to see the whole process, where the primers go, where the Okazaki fragments are, who's going which way, and so forth. And that one ends right here. We're done with Part 1. All the DNA, all the leading and lagging strands and the primers are complete. But now we got to get rid of the primers, replace those with DNA, and then we have to seal those phosphodiester bonds, right? Because if you take your DNA out of G2 and you do that melting exercise, all you get is 2 long single strands per chromosome. There aren't little chunks all over in your chromosome. They're all phosphodiester, all gajillion, trillion base pairs are phosphodiester bonded all the way along. There's no gap. There's no RNA and no gap, so we have to take care of that. All right, let's see what okay. So the PowerPoint pictures are essentially what we've already said. Again, we can think of, in this picture, they're just not showing us the other side. Right? This would be the origin. This would be crotch. Then there's another crotch. Bidirectional, always going the other way, right? Okay, so they're just telling us, right, we add the primer, and if we add in our own polarities, yes, they're going the correct direction, right? Adding to 3' ends, it's anti-parallel. If this strand is going towards the crotch, which strand is it? Leading. Which means, right, the other primase would be here, setting up its primer, then DNA would go that way. And kicking out, remember the stupid single-stranded binding proteins we never draw in there, but they're there keeping the strands apart so that everybody can do their job. And really on the leading side, they're not that necessary because you're really just following right behind the helicase. But on the lagging strand, right, there's that gap between opening of the crotch, adding the primer, and coming back. So that side really needs those proteins there. Okay. So this kind of thing is what you're going to see on the exam, right? And it'll be set up with some sort of polarity, not necessarily, right? But what are the options? Either this way or the other way, right? And then I can tell you either draw on this side and fill it in or draw that side and fill it in. So if you do all 4 possible things, there's nothing new that can show up on the exam, right? So if you practice drawing all of them, those are the only 4 things I can do. Right? There's no other polarity and there's no other side that exists. So make sure you draw your... And the only thing I wanted to add is when you're doing your drawings, one thing, which is why I'm going to provide this template, because, right, when we show, right, we're trying to make it look accurate, right? Right, there's your bubble, there's your origin. And so then a lot of times students will draw their bubble like this. And then they'll go 5' to 3' and 5, oops. Yeah. And, 3' to 5', but then get mixed up, which, right, they're looking at the top, right here, instead of following the strand to here. And so I used to tell students, don't do that. Just make your bubble not cross. Or just draw it like that, right? Now you can't get confused. You know down here it crosses, right? But I'm going to give you that, because then still somebody always does it. And then I feel terrible because they did it all backwards. And then if they had some mistakes in there, I can't even go, oh, I'll give them half partial credit because they messed up. But then there's other things wrong, and pretty soon, a 20-point question, they get 1. Like, don't cross the streams, right? Keep them separate. So I'm going to provide that template and just tell you where's your origin, you're doing the right side, you're doing the left side, blah, blah, blah. Yeah. It'll be like just on a piece of paper, and I'll tell you, fill one side, show me at least 3 Okazaki fragments. So you don't need to put 20, but at least 3 so that I know primer, strand, that sort of thing.
Will we have to label this?
Yeah. So they'll be like A through G or whatever, and you can either just write A next to it or the name primer, and it'll be pretty clear what I want: a box versus a line, an arrowhead, label the crotch. Okay, so we already talked about that, right? We cleared up our naked DNA, and I just call it that because people like the word naked. They like to conjure up images. Okay. Crotch and naked all in one slide. All right. And so yes, somebody else in here said that the leading strand is continuous because it just follows into the crotch. The lagging strand is discontinuous because it literally has little tiny chunks, the Okazaki fragments. Okay. DNA polymerase 3 is the bacterial polymerase that does the leading and the lagging strands. Okay, so the main part of replication is DNA polymerase III. In eukaryotes, there's a gajillion polymerases, and they all have redundant functions, so we're not going to learn names of them or what they do. We're going to use the bacterial names, because DNA polymerase III only does leading and lagging, and DNA polymerase I is the guy in charge of removing primers and adding back. Okay, so we're going to ignore eukaryotic names because there's too many of them. Okay. Right, and we already talked about the lagging strand, blah, 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 has to be at the crotch because of the rules. And then here's them showing you, and so this is the snapshot in time, like here was our first snapshot in time, they're showing another snapshot in time, another snapshot in time. But it's really, a continuum. Okay, so this is the same thing that I drew, it's just the book figures that's neater and less crazy. Aha! So now this would be where I would start my tutorial this would be part 2. Okay? Where I was done with my crazy drawing, now we have RNA and our DNA, and we have gaps. We have places where there's no phosphodiester bond. We need to fix that. As it turns out, our good friend DNA polymerase I is in charge of both exonuclease and polymerase function. So it literally is a complex of proteins, quaternary structure. Yes, the final, little word for the final. That has 2 active sites. One that works essentially exactly like DNA polymerase III, right? Can only do existing 3'OH, incoming 5' phosphate. Okay, but it can also chew out nucleotides from the 5' end. So we're just going to take that, and you guys are just going to believe me because we're in 107, and we'll look at the mechanism of the biochemistry of that in 360. So if you're around for 360 this fall, we're going to do a little more of that. But for time and for our knowledge, we're going to go with DNA polymerase I breaks phosphodiester bonds and pulls out the nucleotides of the RNA. Okay? And as it turns out, it does it takes them off the 5' end, and then adds DNA nucleotides using the existing 3' end of the strand on the other side of it. So let's see if I can make this guy make sense here. I'm going to make this go large. Instead of starting all over, I'm going to use their little drawing and add to it. So, right, this has to be the 5' phosphate of that nucleotide. Ooh, can't really see that. Can you see that? No, that's lame. Okay, we'll draw it like this. 5' phosphate. Right, because the yellow is the RNAs, right? These guys are the RNA nucleotides. That was the original primer. These are just showing the ones he's already popped out, right? This one, the 5' end, because then it's anti-parallel to this guy. And so this DNA polymerase I is literally breaking phosphodiester bonds and having them blow out into the wind. At the same time, we know this guy, we'll say that, right, this guy was going this direction. This is the lagging strand next, right, that was closer to the crotch, right, the whole, hang on, let me get another color, right, snapshot in time, that used to be a crotch, over here used to be a crotch, put the primer, come back. And so if this is the 3' end here, right, this must be the 3' end of that nucleotide. Because the very end over here has to be 5' in order for that to be a strand, 5 to 3. Does that make sense? You buy that? Okay. So guess what? As soon as that first nucleotide is popped out, there is a juicy 3'OH just sitting there waiting for the taking. And guess who's sitting right there on top of it? A DNA polymerase. DNA polymerase I. Gets all excited, and now we have a 3'OH dangling, and there's some DNA nucleotides with a 5' phosphate. Oh! phosphodiester bond, clip up, and do hydrogen bonding. So it clips out a nucleotide and adds one in, essentially at the same time as it's moving. All right, so we'll say the active site of the exo, getting rid of, exiting, and the actual polymerase, right? The exo has to be in front, clips it out. Oh, there's 3' OH, phosphodester bond, blah, blah, blah. But what happens after it clips this bad boy? We'll pretend like that's gone. We'll throw white over it. Watch me. Ooh, watch me make it disappear. Okay. What now is this guy's? Oh shit, that's a 5' phosphate sitting there. And if we filled in, right, we'll DNA polymerase fill, phosphodiester bond on the 3' end. Uh-oh. My 3' OH is now sitting right next to 5' phosphate, DNA polymerase, doesn't fit in his active site. He needs help. Who is going to come to the rescue? Here comes ligase, riding in on his horse, grabs those two, in his active site, bang, phosphodester bond. So every single place there's a primer, that's what's happening. So another common misconception is that it's only on the lagging strand. But remember, the leading strand has to start with a primer, because no naked DNA, leading or lagging, always a primer to start. So on the leading strand, there's fewer primers, because it's one primer essentially per origin on the right side, and one primer per origin going the other way. Whereas there could be 10 primers per origin or more on the lagging strand. But we still have that problem at the origin for the leading strand primer, right? Pull it out, oh no, I haven't lagging. Okay? So across the origin, where leading and lagging bump into each other, the primer boink, right? And all the Okazaki fragments. So most standardized tests will just talk about how are Okazaki fragments fused back together, and you'd say ligase, right? But we know any place there's a primer, we have to remove with DNA polymerase 1, refill, and then that last phosphodester bond has to be ligase, because it's not an incoming nucleotide for our good friend polymerase. Yeah? It's a great question. So if you have our blank thing, right, if I didn't, which I will, if I didn't give you polarity, then you'd be stuck figuring it, so you'll say, okay, if I make this 5' and this 3', and this 3' and this 5', first I'm going to check to make sure I didn't cross the streams, right, it's anti-parallel, my template strands, and then I'm going to give myself an origin down the middle. Okay. Now we just use our rules. The primer has to be anti-parallel, we have to add the 3' ends, complementary, and we don't cross the origin. And so then that's where we just play our little, right, we could just do this. Put a primer in all 4 spots. Right, and go, okay, now we just have to make sure they're anti-parallel. So this would be the 3' end, the 5' end, right, that has to be 5'3", right, because we're checking to make sure, right, this is anti-parallel, right, this is anti-parallel, right, so, okay. And then we do the same thing for these guys, has to be 3. So if you screw this up, if you start wrong, like I did, then you're going to be all wrong. So that's the first thing you do is make sure they're anti-parallel. So rule number 1. Rule number 2 is can only add to 3' ends. Right? So we'd be like, if we were going to take a leading strand, right, just like this one, we'd add to the 3' end. Can we add to this 3' end? Ooh, that's our extra 4th rule. Don't cross the origin. So no. Can we add to this end? No. Do that. That negates the rule, only adds to 3' end. Right? Is this a 3' end? Yes. So we can do this. So that tells us right there that this is wrong, and this is wrong. Well, if it doesn't belong there, then it has to be at the crotch. Point. Right? This is 5 to 3. Add to 3' end. Yep, don't cross the origin. This is 5 to 3. Add to 3' end. Don't cross the origin. And in this case, they can't because it bumps into a prime. So whatever setup you're given, you don't have to memorize, but you have to memorize the rules. Those 4 rules, you can't go wrong. Yeah. Yeah, so because really, like this box represents nucleotides. And so, right, each nucleotide has a 5' or 3' or 3' or 5'. Yeah, I'm just calling this as open. Yeah. Yeah, and that one in the exam is like they make the bubble better. They made a little tiny. I just made mine too big. Yeah, because this will show it complementary if we look back at this. They're stuck together. So it'll be clear that that's still the helix together versus where we go. And then for the lagging, right, you want to have 3. Okay. All right, let's see, what else am I missing? Okay, so this is more, right, we just talked about that. Okay, this is Dr. Stein's deal. We don't need, we never talked about that guy. We didn't talk about him. She loved the clamp loader and the sliding clamp, but we didn't talk about them together. This is cell bio. Okay, so that's, yeah, those terms we don't have to know. The rest of them are all the things we already talked about sort of all together, right? And this would be where the origin is here. And so they're only showing one side. Really, the other side looks exactly the same. Okay, that was, okay. Yes, like this. So this will be the picture that you see for the exam, right? So then, you know, you start here, and you can do your process of elimination, whatever. I'm like, oh my God! Right, so we'd have, right, so this would be where the crotch begins there, right? Okay, that was practice. Oh my God. I should have made you guys, I forgot to make you guys practice. All right. We're out of time, so we're not doing this. We're not doing this. No, we're not doing this. No, we're not doing that. Okay, we're not doing those. We'll revisit that a little bit after the exam really quick, but we weren't going to do mechanism anyways, because I want you guys to take the practice question exam quiz that's in today, which I have no idea what the password is. Let me look really quick. No? Okay. Oh, okay. Yeah, because I don't care if I want everybody to do it. I set it up, so I'm guessing no. But I don't know. Try it either way. Tell me what works. Sorry. I completely forgot about respondents when I was futzing around with this. And so this one you'll be able to review and look at over and over and, you know, use it forever. And then you betcha. Unknown error. Error, Will Robinson.
Is he correct next to it? And it says the correct answer is the one I chose. So
Oh. Did I not put a...
it says where it says incorrect.
But it does say it's correct. Like you get the grade.
Oh, because
Yeah.
two are the same.
No, they're not.
No, they're not exactly the
It
same.
is the correct answer.
Yeah, it's the correct answer. Yeah.
Okay, if you got a green check, you're good. I changed an old question and I changed which answer was correct, so I think that was in the feedback and I didn't delete it or something. So if you get a red X, it's wrong. You get a green check, it's correct, even if it says incorrect, because that was just me in a hurry. Okay, and then before you go, we'll do one Moodle upload as homework whenever this weekend before the exam. So just what I have there. You're just going to fill in the bubble, and you can choose your own polarities for the template. You know, your 5' to 3'. And then I'm going to open one of the uploads on today that, yeah, it'll close Sunday at midnight or close to there, if I can figure out how to do it. You can do either or both just for practice. And if you want to do two bubbles, one with one polarity of the template and one with the other, like for your, only upload one of them, but, oh man, it's logging me out. And then questions. Anyone? Yes, I think. So there was two different practice questions, so some of you got one and some of you got the other. Let me change the time on this so I don't forget.
That's correct, right?
Yeah, so that was in the feedback, which,
Okay.
I changed, whenever and then I
Right.
didn't, yeah.
Okay, I was like.
So green check is correct, even if it says incorrect, that's just me not changing everything. What is the date? What's the date Monday? Monday is the 11th, right? Okay. Hi, what can I do for you?
For the RNA primers,
Mm-hmm.
stay there until, or do they fall off?
So the primates really, so the whole thing is really one big, huge complex. But if we just think about for the leading strands,
Yeah.
that's easier. So the primate is part of that whole complex, so we can think about it going away.
Yeah.
But in the real world, it probably just stays there.
So is there ever a time like for the Okazaki where there's like just a gap present, or is that
So very, very transiently there is a gap. So really, the lagging strand is like the whole complex and the polymerases are all together for leading and lagging, and the lagging strand is fed this way.
Okay.
But there is a dead period between, because otherwise the primers would be too close together and be a waste of time. So it literally does take longer